Okay, who is this Gregor Mendel? I'm sure you've heard his name before, right? When you were in school. Ada tak your, you apa ni? Belajar pasal Gregor Mendel.
Ada.
Kan?
Ada.
Alright. Okay, he was an Austrian monk. Okay, apa yang dia tak puas hati? Do you guys ingat tak apa yang jadi? Apa yang dia tak puas hati? Tengok ni gambar dia. Ada yang dia tak puas hati, something. Ingat tak?
Pokok dia tak sama.
Okay. Ada lagi, before that lagi. Sebelum dia carry out the experiment. Tak ada? Okay. Masa dulu, awal sebelum dia start dia punya eksperimen, that time, there was no concept of meiosis and mitosis. Tak ada mikroskop. There was no knowledge of cells and chromosome, nothing masa time tu. Okay, tapi at that time, there was the blending concept of inheritance. What does this mean? Orang dulu-dulu, diorang percaya if you cross a red and a white bunga, you will get pink. Setiap kali you cross a red and a white flower, you will get a pink flower. Okay. Parents of contrasting appearance produces offspring of intermediate appearances. Okay, ni what they believe. Tapi what happen over time? Contohnya yang pink flower ni, bila dia breed dengan white, jadi apa? And then kalau tinggal lama-lama over time, lagi banyak pink jadi, if you cross pink and pink, jadi apa? So the variation will decrease over time. Tetapi what happen kalau tiba-tiba dah banyak generation, tiba-tiba keluar balik merah and putih. So they could not explain that. The only thing that they could explain, jadinya merah and jadinya putih balik is because they said there was an instability in the genetic makeup, which is wrong. Well, Mendel proved it was wrong. Okay, so Mendel ni, he wants to prove to everyone that there is something else. Bukannya it just the simple step of just blending the two process tu dapat an intermediate. So that's why he carried out the experiment on inheritance of simple traits with the garden pea. This experiment disproved the blending hypothesis, which is the proof that yang blending hypothesis ni salah. Okay, disprove the blending hypothesis. So Mendel's findings were in contrast with this kan. So he proposed the particulate theory of inheritance. Ada tiga, tapi two main one that you have to know is law of segregation and law of independent assortment, which involves the reshuffling of genes from generation to generation. Okay. So these are the three laws that he proposed. The first one is law of segregation, which is during gamete formation, the alleles for each gene segregate from each other so that each gamete carries only one allele for each gene. Gamete. Okay, gamete carries only one allele. So yeah, bila dia kata the alleles for each gene segregate, so this is during anaphase. Okay. So the next one, law of independent assortment. Genes for different traits can segregate independently during the formation of gametes. So most of them believe that this happened during metaphase 1. Okay, and the crossing over occurs in prophase 1. So the last one is law of dominance. Law of dominance is some alleles are dominant while others are recessive. So an organism with at least one dominant allele will display the effect of the dominant allele. So what does that mean? Contohnya kita ada heterozygous. Capital A, atau contohnya tinggi, a trait for tall. T kan, T besar. Okay, T besar, you get a homozygous dominant. Okay, so that person will have, macam mana saya nak ambil ni kejap ya. Ada your friends nak masuk. Kejap. Alright. Okay, I repeat that again. T besar and T besar is homozygous dominant. T besar, T kecil is heterozygous. So as long as ada one dominant allele there, dia dikira it will display the effect of the dominant allele. So that is law of dominance. Okay. Alright. So Mendel worked with the garden peas. Kenapa garden peas? Sebab masa tu garden pea was very easy to cultivate. And then masa tu there was abundance of garden pea. Can be apa ni? Boleh kutip tepi jalan pun boleh. Maksudnya scientists mostly time tu dia orang neglect, dia orang macam take for granted all these garden peas. Sebenarnya garden pea ni Mendel believe that they can be used as an experiment punya sample. So he decided to use that. He decided to use it because the peas are normally self-pollinating, but can also be cross-pollinated by hand. Contohnya dia ambil daripada enter bawa ke stigma from one tree to from one pea to another pea. So dia senang, paling senang nak cultivate. So the pollen was transferred from the enter of one plant to the stigma part of another. Okay. And there's also true breeding that is available. Maksudnya true breeding ni either a homozygous dominant ataupun homozygous recessive. True breeding ni you tengok dia betul-betul purple, dia tak ada the mixed colour. Okay. And then he studied inheritance of relatively simple and discrete trait. So dia pilih trait yang senang nampak. Yang tak adalah terlalu apa ni? You have to examine betul-betul ke apa. This one you can see with the naked eye. Because masa tu tak ada microscope or anything lagi. Okay, bear in mind that. So he chose seed shape. He chose seed color and also flower color for his experiment. Okay, next. So this is what he did. He cut away the anthers, brushing on pollen from another plant. Okay, so all the peas are yellow when one parent produces yellow seed and the other parent produces green seed. Okay, so they cross yellow and green, dia dapat yellow. Dia tak dapat, kalau gabung green and yellow, you would expect to get a blue kan? Betul tak? So he didn't get a blue. He got all peas are yellow. Betul? So out of the observation, the offspring did not possess intermediate characteristics but rather similar appearances to one of the parent. Meaning, katakanlah yang blending theory sebelum ni, yang orang dok kecoh sebelum ni, they said that if you cross the parent, you will get an intermediate characteristic. Tapi from his experiment, he proved that it was wrong. If you would have crossed yang yellow and the green, kalau dapat betul lah blending theory ni betul, he would have gotten a blue pea. But no, he got all the peas were yellow. So he said that the offspring carries the characteristic of one of the parent. That was what he hypothesized from this experiment. So contohnya the offspring could only be yellow or could only be green. Okay, that's why he disproved the blending concept theory. Okay, he proposed the particulate theory of inheritance. Okay, so this is part of the particulate theory of inheritance, which is the first one is law of segregation. Mendel performed crossbreeding experiments. He used true breeding, which are homozygous plants. He chose varieties that differed in only one trait. Contohnya satu eksperimen tu dia carry out untuk seed color je. One more experiment dia carry out untuk flower color. Okay, only one trait. He performed reciprocal crosses. Okay, this is what he did. He started off with the parental generation. He labeled that as P. And then the first filial generation, which is F1. And then the second filial generation, which is F2. So then he formulated the law of segregation. Okay, so let's look at these crosses. So if we use true breed ataupun purebred one, homozygous kan? So T besar, T besar ataupun T kecil, T kecil. So for this example here, T allele stands for tall plant. T, small t stands for short plant. Okay, this is the step that you have to know to answer your exam kalau ada any crosses. Okay, you have to label the P generation. Okay, out of the P generation, you take out the P gamete. What are the possible P gametes? So T daripada T besar, homozygous dominant, you take out T besar. So homozygous recessive, T kecil, you take out T kecil. Okay, and then the crossing occurs, fertilization. So these are T, big T gamete, small T gamete, gabung, dua, fertilized, what do they get? So F1 generation will get a heterozygous, T besar, T kecil. Okay, and then it self-pollinate, okay, F1 gametes to get F2. Okay, so F2, what do you get? So you do a Punnett square, macam ni. Your cross, you take out the gamete. T besar and T kecil. So you put the gametes here. One for the female and one for the males. So you cross senang. T cross with T, you get T besar, T besar. T besar cross with T kecil, you get T besar, T kecil. And then same for the other two. So from here, you get the genotype. Okay, these are the genotype. But what are the phenotype? So if you see here, kalau T besar, it will be tall. The plant will be tall. If it has a T besar, T kecil heterozygous, the plant will also be tall. Remember the law of dominance. Okay? So tall plant here, this is a tall plant here, this is a tall plant here, and then a small t, you get a short plant. So out of this monohybrid cross, you would expect to get a ratio of 3 to 1. Okay? 3 nisbah 1, 3 to 1. Okay, 3 tall and 1 short. So this is monohybrid cross. Okay, so this is his result. Annoying lah this thing. Cuma saya nak turunkan. Okay, never mind. Alright. So, for the stem length, he observed there were tall and there were short characteristics. So, daripada ni, dominant dia ada 787 and recessive was 277. So, if you see the ratio, it was 2.84 to 1. Okay, he carried out lagi on pot shape pula. So, pot shape, whether it is inflated ke or it is constricted. So, he got these numbers. And he did the ratio, it was 2.95 to 1. Okay, he repeated again all the experiment. Kali ni guna seed shape pula, and then seed color, and then flower position, flower color, and lastly the pot color. Okay, so he observed, if you see all the ratios dekat sini, mostly you can conclude that it is a 3 to 1 ratio. 3 nisbah 1. 3 dominant, 1 recessive. Okay, so these are monohybrid cross with only one trait. Okay. So from the experiment, 3 to 1 ratio among F2 offspring was possible if the F1 parents contain 2 separate copies of each factor. One is dominant and the other is recessive. So bila parents ada 2 separate copies, which is T besar ke T kecil, you would have gotten the 3 to 1 ratio. Another hypothesis was the factors separated when the gametes were formed and each gamete carried only 1 copy of each factor. Gamete, kan? Gamete tadi only 1 copy of each factor. Bila fertilized, baru jadi another factor. So random fusion of all possible gametes occurred upon fertilization. So from here, he concluded the law of segregation. Okay, so this result of the monohybrid cross all F1 plants were tall. So this disproved the blending hypothesis. So katakanlah tadi yang tall and short tu. Kalau blending hypothesis antara tall and short parent, you would have gotten an intermediate punya apa ni? Height. But no, he got all of the F1 plants were tall. So this disproved the blending hypothesis. Okay, so next we go on into the terms that you have to use bila you explain. So locus, what are locus? Locus is this. Macam satu locus dekat tempat sini. This is another locus. This is another locus. And this is another locus. So that each is a particular location where the genes occur. Okay, so what are alleles? Alleles. So these are allele. This one allele. These are alleles. So this G besar G kecil ni adalah alleles at a gene locus. Okay? So dominant alleles. Dominant, if you see G besar is a dominant allele. G kecil is a recessive allele. So dominant alleles, one allele masks the contribution of a second allele at the same locus. Contohnya G besar and G kecil. So you will still get a dominant. Okay, heterozygous, but it is dominant. Okay, recessive allele. A recessive allele only shows if the individual has two copies of the recessive allele. So to make it recessive, it all has to be G besar, G kecil. Okay, only then it will become a recessive. Okay, what are genotype pula? Genotype and phenotype. So genotype refers to the two alleles an individual has for a specific trait. It can be homozygous and it can be heterozygous. Okay, like this. Contohnya lah. Okay, A besar, or A kecil, or it can be A besar, A kecil. Okay, what are phenotype? Phenotype refers to the physical appearance of the individual. Okay, apa yang you nampak. Genotype yang you tak nampak dalam badan, contohnya T besar, your phenotype is tall. Kalau T kecil genotype, your phenotype is short. Okay, don't confuse between genotype and phenotype. Okay. Alright, alleles. So alleles for each trait in a pea plant is controlled by two alleles. Okay, two alleles. 1 and 2. So dominant allele, capital letters, masks the expression of the recessive allele, which is lowercase. Okay, contoh macam ni. B besar masks the expression of the recessive allele. So it will be still dominant. So alleles occur on a homologous pair of chromosome at a particular gene locus. Locus dia katakanlah B besar ni dekat atas sini, it will still be homozygous dominant. Regardless dia ada dekat gene locus yang mana satu. Because it can, this is a part of variation. Sometimes it can occur here for your parents, tapi you, dia dah sampai sini. So those are the factors for variation as well, for diversity. So homozygous are identical alleles and heterozygous are different alleles. Okay. So Mendel's cross. Okay. So what do we know by this Mendel's experiment? Okay. So what we know is dominant and recessive alleles represent DNA sequences that code for protein. Okay. So your parents DNA sequence dengan you punya DNA sequence lain ke sama?
Lain.
Lain. Why? Because ada variation. But apa yang sama is your parents are human. So they bring the coding untuk human as well to you. But your traits are different between your parents and you. Parents you katakanlah your mom have straight hair, your dad ada curly hair. So sampai ke you, tiba-tiba jadi wavy ataupun jadi curly. Tak ikut your mom, ikut your dad. Okay, so those are variation. So but the DNA coded from the fertilization of gametes from your parents, gamete mom you dengan gamete your dad fertilized, they contain the DNA sequence that works for that will encode protein for you, for your body to carry out the function. Okay, faham? Faham tak? Okay,
Faham.
alright. The dominant allele code for protein associated with normal gene function within the cell. Okay, dominant selalunya kita cakap it is okay, normal. Selalunya lah, okay. So, recessive allele pula represents a loss of function. Tapi tak semestinya juga sebab dimple. You think it's a dominant ke recessive?
Recessive.
Dominant.
Recessive.
Yang mana cakap dominant, Google dulu, okay. Sama ada is dominant ke recessive. So you would know, sometimes orang kata recessive ni teruk sangat. No, sometimes recessive ni can be macam, we call it as a, kalau dalam bahasa Malaysia penyeri lah kan. Those yang ada dimple ke apa, you will look sweeter contohnya. It's not all bad. Bila kata loss of function ni, it's just loss of some cells in the pipi. That's why jadi dimples. Okay? So during meiosis 1, homologous chromosomes separate and two alleles separate from each other. So this explains the law of segregation. Two alleles separate from each other ni, this is anaphase. Okay? Okay, so next kita dah habis dengan law of segregation. Now we go on into the second law, which is law of independent assortment. Okay, law of independent assortment ni usually refers to dihybrid crosses. What does it mean by dihybrid crosses? What do you understand by dihybrid cross?
Two traits.
Yes, betul, two traits. Kalau monohybrid is one trait. Dihybrid, two traits. Okay? So, Mendel buat eksperimen lagi sekali. Kali ni guna two traits which are dihybrid crosses. So, he started with true breeding plants differing in two traits. F1 plant showed both dominant characteristics. So, F1 dia dapat dominant. And then F1 self-pollinated, and then he observed the phenotype among the F2 plants. So let's see what happened. So he hypothesized two things, dua possible result. Okay. So if dominant factor dia T besar, G besar, TG, always segregate into F1 gametes together, and recessive factor, which is a small t and a small g, always stick together. So he would only expect to get only two phenotypes je. Okay. Another one hypothesis is if four factors segregate into F1 gamete, then there would be four phenotypes, four types of phenotype. Okay. So from his experiment, the results supported the second hypothesis. For dihybrid cross, akan ada four phenotypes. Okay, for phenotype among F2 plants. So he formulated the law of independent assortment. Okay, let's see the detail on the crosses. Okay, so this is the cross. Nampak? Macam nak saya nak alihkan benda ni. Okay, so dihybrid cross done by Mendel. What did he do? He crossed, okay, as I've mentioned before, what you need to have for your exam mesti tulis the P generation dulu. P. Okay, and then after the P generation comes the P gamete. Turunkan gamete dia. Okay, so what are the P generation here? Here we have the T besar, G besar, G besar. So this stands for tall plant green spot. Okay, the recessive are the small t, small g, and small g. So these are short yellow. Tengok tak plant dia? Ni tinggi kan? And then green. And this is short and yellow. So these are dominant and these are recessive. So comes next is the P gamete. We take out the capital T and the capital G, you cross it with the small t and small g, cross these two gamete, one from each parent, fertilize, akan dapat T besar, T kecil, G besar, G kecil. Tapi what are the phenotype? What is the phenotype here? Can anybody tell me what is the phenotype for F1?
Tall green.
Yes, tall plant and green pot. All of it pula tu. Semua F1 dapat tall and green pot. Okay, so then comes the self-pollination. F1 cross dengan F1. So, to make it easier, he did it in a Punnett square. Okay, so the female, dia keluarkan the possible gamete. What are the possible gametes? Possible gametes, sorry. So you get a T besar, G besar, T besar, G kecil, T kecil, G besar, and T kecil, G kecil. So these are the possible gametes. Okay, this is for the sperm and these are for the eggs. So sama juga for the sperm. So this is for the eggs. For the sperm, you take out the possible gamete. So this is what you will get. And then you do the crosses. Semua you cross. And finally, if you see the ratio, you would get a 9-3-3-1 ratio. 9 plants that is tall and green pot. 3 plants that is tall and yellow pot. 3 plants that is short and green. And 1 that is short and yellow, which is only 1 recessive. Okay. So this produce offspring with 9-3-3-1. Okay. Anybody ada yang confused ke dengan cross ni? Anybody nak tanya apa-apa? Oh my god, can you hear that? Kucing pula bergaduh. Okay. Right, so if no questions, I will proceed. Okay, so back to the independent assortment and segregation during meiosis. So this part here is where the parent cell has 2 pairs of homologous chromosome, and then all orientations of homologous chromosomes are possible at metaphase 1, keeping in the law of independent assortment. So ini just a refreshment balik. Then at metaphase 2, each daughter cell has only 1 member of each homologous chromosome, keeping in pair with the law of segregation. So lastly, so these are the possible combination of alleles that occurs in the gamete. So this is suggested by the Mendel's 2 law. Dia gabungkan the independent assortment and also the law of segregation. So Punnett square, tadi saya ada sebut pasal Punnett square, right? So it was named after this fella here. He is called Reginald Punnett. Okay, so this diagram is used by biologists to determine the probability of an offspring having a particular genotype. Okay, so dia letak 1 possible gamete here, another possible gamete here, and also the gametes from another parent. So seorang satu dekat sini and the other one down here. And then you do the crosses. So kalau dalam exam, I would recommend you to use a Punnett square rather than doing the crosses banyak-banyak, because sometimes you can get confused. Sometimes it can be too much of the detail that you get confused. Contohnya you nak buat sampai the law of independent assortment, dihybrid cross punya cross kan, it be too much of process. So it's better that you do it in the Punnett form. Okay. So all possible sperm genotypes lined up on one side and possible egg genotype are lined up on one side, and all the possible zygote genotypes are placed within the squares. Okay, so contohnya macam ni. This is a Punnett square. Untuk allele dia unattached earlobe if capital E and a small e is attached earlobe. Okay, so these are the parents. So you bring down the possible gamete. So possible gamete for the father is E besar, E kecil, and the possible gametes for the mother is E besar, E kecil. Then you do the crosses. Okay, bila you do the crosses, what you can get is siapa yang dapat E besar, the probability is 1/4. Okay, for E besar, E kecil, the probability is 1/4 as well. Tapi ni kenapa? Okay, sama. 1/4 as well. And then the chance of a small e and a small e is also a quarter. Cuma, what are the phenotype ratio? The phenotype ratio is 3:1. So, 3 ada unattached earlobes and 1 with attached earlobe. Okay? And the sum rule allows us to add genotypes that produce identical phenotype to find out the chance of a particular phenotype. Contohnya yang macam ni. 1/4, 1/4, you can write that as half. Also, okay? So, 1/4, E 1/4, E besar E kecil, 1/2, and E kecil 1/4. Okay, let's watch a video on the dihybrid cross. Kejap. This thing. Kena pergi dia punya tu. Dia punya play button. Sekejap ya. What happened? Okay. Share. Okay. All right. Okay.
In many ways, a two-trait or dihybrid cross is
Can
very
everybody hear?
similar to a monohybrid cross. The key to understanding a dihybrid cross is to first remember that the traits in the cross assort independently from one another, meaning that they are on separate chromosomes. The second thing to recognize is that we are looking at probabilities, or the chances of producing offspring with a certain genotype or phenotype. In this example, we are going to be using two human traits, hairline and finger length. Our two alleles for hairline are the recessive straight hairline and the dominant widow's peak. A widow's peak has a characteristic W shape in the center. The two alleles for finger length are the recessive long fingers and the dominant short fingers. In this example, we are going to use an individual who is homozygous dominant for both traits, big W, big S, and one who is homozygous recessive for both traits, little w, little s. These individuals are called the P, or parental generation. Remember that the end result of meiosis in humans are gametes that have a single copy of each trait. The homozygous dominant male in this diagram therefore can only produce sperm with a big W, big S genotype, and the homozygous female can only produce little w, little s eggs. Upon fertilization, the offspring will be heterozygous for both traits, or big W, big S, little s. This is called the F1, or filial generation, since they are the first generation of offspring following the parental generation. To generate the F2 generation, we are going to look at a random mating between two individuals from the F1 generation. Before we take a look at the F2 generation, let's take a look at the possible gametes that may be produced by the F1 generation. Here's where the dihybrid cross gets a little tricky. During meiosis, homologous pairs of chromosomes assort independently. Since the big W and big S genes are located on separate chromosomes, it is possible that the big W allele can be paired with the big S allele, or the big W can be paired with the little s. The same is true for the little w allele. It can pair with either the big S or the little s. Therefore, we end up with four different options: big W big S, big W little s, little w big S, and little w little s. Here we see a Punnett square that lists the possible combinations of gametes for each parent. Within the Punnett square are the possible offspring from this cross. The first thing you should notice is that there are 4 different phenotype combinations, but 16 different combinations of genotypes. Each of the phenotypes is based on the combination of alleles that were received from each of the parents. Let's take a closer look at how we can use probabilities to find the same results. The important thing here is that you recognize that you don't need a complex Punnett square to do these crosses. Instead, we can use the basic laws of probability. Since the W and S genes are on separate chromosomes, we can treat their inheritance as separate events. For the W gene, if the parent is heterozygous, 1/2 the time they will contribute a big W allele, and 1/2 the time it will be the little w allele. Therefore, 3/4 of the time the offspring will have the dominant phenotype with regards to the W gene, and 1/4 of the time it will have the recessive phenotype. The same thing will happen for the S allele. The probability of an offspring having both the dominant W and S phenotypes is equal to the product of the individual probabilities. In other words, there is a 3/4 chance of having the big W phenotype and 3/4 chance of having the big S phenotype. 3/4 times 3/4 equals 9/16. If you count the number of boxes that are dominant for both traits, you get 9 out of 16, or 9/16. On a similar note, the probability of having an individual who has recessive phenotype for both traits is 1/16. If you multiply the probability of receiving both a recessive little w and little s allele from each parent, you get 1/4 times 1/4, or 1/16. Overall, we can see that the phenotypic ratios produce a 9:3 to 3:1 pattern of inheritance.
Okay. Nampak. Oh ya, laptop saya hari ni dia buat pasal sikit. Sekejap. Hmm. Stop pula. Kejap ya. Start share. Share screen. Okay. So back to here again. Saya lupa cakap tadi when I said the chance yang ni E is a quarter. And then a chance for E besar dan E kecil is half. And then E kecil is a quarter. So for phenotypic ratio, you would get a 3 unattached earlobe sebab it is all 3 atas ni is dominant. Here. Walaupun it is a heterozygous, but it still mask the recessive punya allele, so you will still get a dominant. So you will get 3 unattached earlobe and 1 attached earlobe. So this is the phenotypic ratio. Okay. Kalau exam selalu dia akan tanya either the genotypic ratio or the phenotypic ratio or both. Okay. So next we go on into test cross. Okay. Test cross. Test cross, individuals with recessive phenotype always have a homozygous recessive genotype. Okay, selalunya kita buat test cross ni to know whether the gametes from the parent sama ada homozygous dominant ataupun heterozygous parent. Okay, contohnya kalau dalam ni, Y besar, you cross with an unknown, okay, you cross dengan a homozygous recessive. Test cross mesti dengan homozygous recessive. So these are gametes from parent of unknown genotype and these are the gametes from recessive parent. So this cross resulting in all dominant offspring indicates that the parent is homozygous dominant. Okay? So if you cross Y besar and a Y kecil, you would get this. So what happened down here? If Y besar and null allele and also cross dengan recessive, kalau dapat macam ni, you would know that this is a 1:1 ratio. Bila 1:1 ratio, it is usually a heterozygous parent. So a test cross resulting in a 1:1 ratio of yellow to green offspring indicates that the parent is heterozygous. You have to know test cross, dia punya nisbah is 1 to 1. Okay? Right. So this is an example of a one trait test cross. Okay, contohnya dekat atas ni, we use only is either a tall and a short plant. Dia punya height is the trait. Okay, so we have the parent as a T besar, T kecil and cross with a recessive. So test cross, you always have to cross with a homozygous recessive. With a T kecil and T kecil. So what do you get? You get you bring out the possible gamete dulu, T besar, T kecil here. And the possible gamete from here, which is T kecil. So this is what you get. Okay, the ratio is 1 to 1. One tall and one short. Okay? One tall and one short. So, this is an example B pula. T besar, T besar. Yang tadi T besar, T kecil. Okay? So, kalau T besar, you cross with a homozygous recessive untuk test cross, you would get all of the phenotypes are tall plant. Okay? This is not a 1 to 1 ratio. Kalau heterozygous, you will get a 1 to 1 ratio. Okay? Okay. So crossing both with recessive plant can show the result. For example, a heterozygous cross with the homozygous recessive will produce offspring ratio 1 to 1 of tall and short plant. And then if you cross the homozygous dominant with the homozygous recessive, it will only produce tall plants. Okay, so this is a one trait test cross. So next we have the two trait test cross. So kalau two trait test cross, what do we get? We get the, okay, P generation dia, you get L besar, L kecil, G besar, G kecil. Which stands for long wings and grey body. So you cross with the short wings and black body, which is L kecil, G kecil, G kecil. So what do you get here? You would expect to get the ratio of 1, 1, 1, 1. Sama rata semua. 1, 1, 1, 1. If you cross it with a homozygous recessive. So this is a test cross. Test cross, you will always get a 1 to 1 atau kalau one trait atau 1 to 1 to 1 to 1. Kalau two trait test cross. Okay? So katakanlah kalau you cross, you punya parent ni ialah a homozygous dominant. You cross with a homozygous recessive, will only produce L besar, L kecil, G besar, G kecil offspring. So you will get a long wing and a grey body. Okay, so for this one, you cross a heterozygous parent with a homozygous recessive parent, it will produce the offspring ratio of 1, 1, 1, and 1. Okay? So if the individual with the dominant phenotype is heterozygous for both traits, then the expected phenotypic ratio is 1 to 1 to 1 to 1. Always remember test cross 1. Kalau monohybrid cross 3 to 1. Maksudnya 3 nisbah 1. Kalau dihybrid cross 9, 3, 3, 1. Kalau test cross 1, 1 ataupun 1, 1, 1, 1. Okay? Alright, so next we go on into the next subtopic, which is Mendelian pattern of inheritance and human disease. Okay, so genetic disorders are medical conditions caused by alleles inherited from parents. Autosome. What are autosomes? So autosomes are any chromosome other than the sex chromosome, other than the X or Y. Okay. So genetic disorders caused by genes on autosomes are called autosomal disorder. Autosomal disorder ni can be either dominant or it can be recessive. Okay, what does it mean by autosomal dominant and autosomal recessive? So if the individual has an A besar disorder and has the disorder, so maksudnya dia akan ada the disorder. Pening tak? Pening. Okay. As long as you see a dominant allele. When you see a dominant allele, it has the disorder. Okay. Kalau yang ni, A besar, A kecil, which is a heterozygous, dia ada the disorder. Sebab it is autosomal dominant. Kalau it has an A kecil, which is homozygous recessive, it does not have the disorder, doesn't have the disease. So those yang ada homozygous dominant and also heterozygous, dia yang can get the disease. Okay, that is for autosomal dominant. Kalau autosomal recessive, an individual with A besar, which is autosomal homozygous dominant, does not have the disorder. Dia tak akan dapat resisto. Dia tak ada. So an individual with a heterozygous does not have the disorder because they still ada the dominant allele. So to get the autosomal recessive disorder ni, it has to be both homozygous recessive. Okay, so let's go into detail on that. Okay, contohnya yang ni. The first one is the autosomal recessive pedigree. If you see here, gambar yang ni, A kecil is affected. Okay, and A besar, A kecil is a carrier. Carrier which is dia unaffected. So A besar unaffected. Only yang affected is mana yang ada homozygous recessive. So contohnya parent homozygous recessive. The cross with a dominant, regardless dia dia heterozygous ke dia homozygous. So those anak dia yang ada dominant allele tak dapat the disorder. Only those yang ada homozygous recessive will get the disorder. Okay? Right, so contoh disease yang selalunya for autosomal recessive is the methemoglobinemia. Yang ni yang dia jadi, your tangan akan jadi bluish purple, dia macam lebam, macam tak cukup darah tu. Okay, selalunya yang ni jarang lah kita dengar, tapi it's harmless. Okay, so another one is cystic fibrosis, the accumulation of mucus dalam your lung atau dalam your pancreatic ducts. Okay, selalunya akan jadi dekat bronchial tubes kita lah, dalam lung kita. So this is another example of autosomal recessive pedigree. Okay, I dah ada nak betulkan sikit in your medal textbook. So if you refer on page page 196. Okay, dekat bahagian autosomal dominant disorders, dia punya ayat bawah tu tiba-tiba jadi autosomal recessive disorders are typically the result bla 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 bla. Tajuk dia autosomal dominant, tapi dia explain pasal autosomal recessive. So please avoid that lah. Just jangan baca the first paragraph. The second paragraph dah okay when it comes to a number of autosomal dominant ni. Okay, page 196 of your medal textbook. Dia dah tersalah copy balik because if you see on page 195, ayat dia sama. Bawah the topik ni, autosomal recessive disorder. Okay. This part here. Nampak tak? Okay, page 195. Betul. Page 196 yang salah. Yang autosomal dominant disorder tu, the first paragraph. That one dia tersalah pula copy yang jadi autosomal recessive disorder. Right? Okay, so this is the disease methemoglobinemia. Dia jadi biru, bluish purple. Okay. And then this is the cystic fibrosis yang mana dia punya ada defektif dekat dia punya chloride ion punya channel. So a lot of the sticky mucus akan ada dalam lung kita. Okay, so next one is the autosomal dominant pedigree. Okay, dominant. When it comes to dominant, you have to remember yang besar, as long as ada the dominant allele, it is affected. Okay, dia tak ada carrier, dia terus affected. Okay, so autosomal dominant ni sangat bahaya sebenarnya. Kalau you dapat the copy, even though it's a heterozygous, you will get the disease. Okay, so yang contohnya kalau you tengok ni, A besar, A kecil heterozygous, it is affected. Sama, okay, affected. Yang tak affected only if a homozygous recessive. It is unaffected. This is called autosomal dominant. Okay. So contoh autosomal dominant, we have the osteogenesis characterized by weakened brittle bone. Okay, another one is the spherocytosis. Yang ni apa ni? Red blood cell dia becomes fragile and burst easily, which is very rare lah kita dengar. Okay, spherocytosis. Okay, selalunya when it comes to the question of autosomal dominant recessive, kita akan bagi kalau dalam exam, contohlah kita akan bagi the parent je. So you do the crossing. What are the percentage of having affected ke unaffected allele? Okay? And then sometimes what you have to do, you might have to know a bit on the disease. Disease mana yang autosomal dominant, disease mana yang autosomal recessive. Selalunya disease yang ada dalam buku medal je lah. Tak adalah kita ambil daripada luar ke apa.